Hey, Bio 1 students, Mr. Yance again. Let's see if we can go through some more of this photosynthesis process today. So what I'm going to do is go through some notes here and then have some questions for you to answer for your e-learning about the notes, just to see if you paid attention or if you have any questions or anything like that. So I'm actually probably just going to do like a video review like we've done for other YouTube videos, except this time it's going to be my ugly mug on there. So we left off talking about the light reaction, and again, these 4 steps. Hopefully you have this all filled in. The light-dependent reaction, obviously light is required. And we broke it into steps 1, 2, 3, and 4. Understand photosystem 1, photosystem 2, that those were 4 events that took place at the same time, not in sequence. And we basically produced 2 major things. We produced tons of ATP, steps 1 and step 3, and we produce a compound called NADPH2. Okay, and that's what we're going to talk about using some of that today. You'll see where that kind of comes into play today and tomorrow. In the Calvin cycle, which is this side over here, so this is the side we're going to talk about today. It does occur in sequence, so it is steps 1, 2, 3 in order. So that's kind of where we're going to pick up. I'm going to do just a little bit of it today. I probably could do all 3 steps today, but I think it might get a little confusing, so I'd rather do it in little chunks and do it that way. And understand that that all this goes together like a puzzle. So hopefully you'll see, oh, this is how all this fits together. I talked about this the other day. This isn't a magic act. We're not pulling a rabbit out of a hat. Everything has to occur and everything has to be produced and come from somewhere. And we're going to talk about that. So I think this is a good diagram down here too to kind of start with. This is I didn't show you this the other day. This is a diagram I came across. I think shows photosystem 1 and 2 very well. You can see these guys with this big hammer with a photon, big sledgehammer that's going to produce our energy, and that's going to knock this electron up here, and we're going to dump it in the bucket here and make NADPH2, which you will see today. And then photosystem 2 over here, same thing. Photon hits it, same hammer. Electron comes up here. It donates electrons over here to use, and we're going to produce huge amounts of this ATP. So that's kind of where we're at. So what do we do with that ATP and what do we do with that NADPH2? Okay, this is where the other reaction comes into play, and it's called the Calvin cycle. You can see it up there, so kind of highlight that. Light is not required because we're using things we made in the other reaction, so we don't need light. We used to call this the dark reaction. We don't like to call it that anymore because too many students were thinking, oh, okay, this only occurs at night and in darkness. Not true. It can occur during the day as well. So we renamed it. Sometimes you see light independent, which I think is a good word, and sometimes called the Calvin cycle. You can use those words either way. Could be an A and B both question on the test. Good possibility. All right. It's the second stage of photosynthesis. Light dependent, light independent. 2 separate things. And here's a summary. This is going to go on the first tab on the right. It involves taking CO2 out of the air and fixing it in something we call glucose, which is C6H12O6. The waste product is water. A little bit different. Oxygen is the waste product of the light dependent reaction. The light independent, water is the waste product, and it occurs in a different place. We call it the stroma. The light dependent is in the grana. This is in the solution around these poker chips, so we're not occurring in the green part. It's occurring outside here. So those are the big differences. A different waste product, different places occurring, and a different waste product. So we're basically, it's called carbon fixation. We're going to take carbon out of the air and put it in C6H12O6. Okay, so let's jump right in. So put that stuff there, 1, 2, and 3, and what I have in red, put that on your foldable on the top tab. So it's going to go on this one right here. And then we're going to talk about step 1. And a lot of these steps today and tomorrow are like chemical formulas. So you'll see kind of how it all goes together. And I want to take my time on step 1 because I think it's very, very confusing sometimes. So kind of keep that in mind as we kind of go through that stuff, okay? So what happens is a plant sits there and it has this compound called RUDP. RUDP stands for ribulose diphosphate. What you need to know is a 5-carbon sugar with 2 phosphates attached to it. That's what it looks like. So it just sits there and waits. And when carbon dioxide is going to come in, if you guys remember, we talked about this the other day as well, it comes through this opening. So the same opening that allows oxygen in or oxygen out is going to allow CO2 to come in. So it's going to come through these openings. There's a bunch of these, thousands of them on the leaves of trees. Okay, so CO2 is going to come in from the air. That's why we call plants carbon sinks sometimes, because they absorb huge amounts of carbon. And we know how bad carbon is for our environment. We have way too much of it. So that's another benefit of green plants, is they can suck out huge amounts of carbon. And this is a carbon acceptor. Here's where this gets a little bit confusing. So let me explain to you. What we are ultimately after is a compound called C6H12O6, glucose. That's the ultimate food that a plant's going to produce that keeps it alive and that we utilize as well. Okay. Now, we get there with this reaction. Let me show you. Okay, so put this in your foldable. Okay, so we get there. You can see CO2 plus RUDP. You get a 6-carbon sugar, and people are going to look at that and they're going to be like, "Isn't that what we're after?" Yeah, it is. The problem is we don't even give this 6-carbon sugar a name because it falls apart. Not good. We don't want to produce something that falls apart. So that's why we have steps 2 and 3, is this is not the end of the story. Yeah, it's a 6-carbon sugar, but it's not glucose. Okay? And this 6-carbon sugar falls, actually literally splits in half. So don't make this more complicated than it is. It becomes 2-PGA. Not the Professional Golfers Association, but phosphoglyceric acid, we call that. And it's a 3-carbon sugar times 2. Okay? So that's what I want you to put on that first tab. Okay? Now, understand this is a crucial part to this process. Okay? Remember the number 2. So when I go through notes tomorrow, you need to remember the number 2, because that's basically everything we're going to talk about in steps 2 and 3 go through twice. Okay? So you're going to think that the numbers don't add up when I go through this process, but everything's going to work twice because I have 2 PGAs sitting there. One's going to go through steps 2 and 3, and then the next one's right behind it. All right? Now, I could finish up notes, but I think it's going to get really confusing, so I'm just going to stop with notes right there. Really short today because I don't want to make this too complicated. Here's another picture. I want to show you some things. This is occurring out here in the solution, not in these little stacks. It's actually occurring outside here in what we call the stroma. Okay, not to be confused with the stomata. The stomata is the opening on the leaf. A lot of people get those backwards on my test. The stroma is the solution inside the chlorophyll where the light independent reactions taking place. All right, and this is where we're at. So let's look at this color sheet, which should look eerily familiar. Okay, you're going to see ATP come in. You're going to see NADPH2 come in. That's going to come in tomorrow. Where we're at right now is this CO2 is coming in, and RUDP is coming in, and we're going to combine those two and make those 2 PGAs that we're going to eventually convert into glucose. Okay, so that's kind of where we're at. So on your foldable, fill in this top tab on the right. So fill in this tab with that introductory stuff, and fill in this carbon fixation. We'll do steps 2 and 3. Just a little short part of notes tomorrow, and we'll be done with photosynthesis when you come back from break. We'll review everything, and then we'll have a review game, and then we're going to take our test probably about midweek. Okay, it'll be a Google form test like we did first try, so nothing changes there. All right, questions on that? If you do, please let me know. Email me, tell me on Monday, or if you're a virtual student, email me, and I'll try to explain things in a different way if I need to. I'm hoping this makes sense. I don't want any of you to get to the test and say, I don't understand any of it. I've kind of broken it down into chunks for a reason. So kind of watch these videos. The good news is I got videos. You can stop, you can replay them, you can go over them. I give you a lot of homework assignments that relate to these as well. So here's what I want you to do is fill in the foldable, fill in the blank notes, or highlight, I should say. We're highlighting, my fault. Highlight what I have and put that in your foldable, and fill out that little video review, turn that in for e-learning. All right? Hey, have a good rest of the week, and we will talk to you tomorrow. See ya. Bye.